I'm Nishat Anjum Bristy. I'm a PhD student at Carnegie Mellon University from Professor Russell Schwartz's lab. And today I'm going to talk about my work on reconstructing tumor evolutionary histories from single cell sequencing data, incorporating single nucleotide variants, copy number alterations, and structural variants. There is a famous saying by Dobzhansky that nothing in biology makes sense except in the light of evolution. This applies for cancer as well. So cancer is an evolutionary disease where healthy cells acquire series of deleterious mutation over the course of genetic progression and becomes uh, aggressive. This follows the process of natural selection, so the cells acquire mutations which have a selective advantage over the other mutations. These mutations are called driver mutations, shown by the stars in the picture. And these driver mutations expand in the cell populations and create distinct cell subpopulations, also known as clones. This is the clonal theory of evolution in cancer. Now, the order of mutation matters because a driver mutation at an earlier time point will be might be more aggressive than another driver mutation at that time point. And the common technique in the field is to build tumor evolutionary histories to understand tumor progression, uh, where in these trees, each node is a cell or a distinct tumor clone, and each edge would represent tumor mutations that have occurred from the parent to the child. And detecting a mutation in an earlier branch and targeting it in that earlier branch might help the patient to survive. That's why we are building these evolutionary histories. So the mutations can differ in different ranges. For example, single nucleotide variants are one letter changes in the genome. Copy number alterations, or CNAs, are larger scale genomic changes where a genomic segment is duplicated, amplified, or deleted. And structural variants, or SVs, which include insertions, inversions, translocations, and larger duplications and deletions. Now, all these mutations contribute for the tumor to progress. And however, the methods in the field are using only a subset of these mutations. My goal here to use all these mutations to build a full picture of the evolutionary history of how the tumor is progressing. Now, the field is using different types of data sets. For example, bulk sequencing data. It is the most commonly used data set in the field, where each bulk sequencing tumor sample will be a mixture of tumor clones. And the computational problem here is to deconvolve the tumor clones and identify individual clones. This is a computationally hard problem. So with the advent of single cell sequencing, we do not need to deal with this problem because in single cell, we have the genomic sequence of each cell. However, single cell sequencing, it comes with its own errors, such as allelic dropouts, false positives, and false negatives. Now, from these data sets, clonal trees or cell trees are built with numerous approaches, such as probabilistic approaches, combinatorial approaches, integer linear programming, and distance-based methods. I am showing the single cell sequencing and integer linear program because I'm using them in my method. So the problem I'm trying to solve is to build tumor evolutionary histories to understand tumor progression from erroneous single cell sequencing data using all the variant types. My input will be single cell tumor samples where the variants are called, and the output at first will be the tumor clones from the tumor single cells and a clonal mutation matrix. How do I do that? I use the pre-existing method MEDIC2. I won't go into the details, but its inputs are single cell copy number alterations, and its output are pairwise distances among the cells. So once I have the pairwise distances using the MEDIC2 algorithm, I do a hierarchical clustering to find individual tumor clones. And once I have the clones, I take the modes of the mutations of the cells that are mapped to the same clone. Now that I have a clonal mutation matrix that I just explained in the previous slide, which looks like this, where the rows have the clones and the columns are the mutations, the first few columns shown in violet are for structural variants. The next column shown in blue are for SNVs. These two are binary. However, the copy numbers are not binary. They are integers, and we deal with allele specific copy numbers. So this is the input in our second stage, and output will be a clonal lineage tree and an estimated copy number matrix, which is a similar dimension of the matrix I just explained. However, it will be an error corrected version of that one. And we will try. Our goal is to try to minimize the evolutionary distance as well as the this difference between this true or observed versus estimated copy number matrix. Now, as I said before, I'm using the integer linear program. Integer linear program has some constraints, some variables, and an objective function. So I put constraints on the edge on an edge matrix to build the tumor phylogeny uh, or tumor evolutionary history, where edge matrix we represent edge matrix by E. I put constraints such that the root of the tree is a normal clone so that no mutation happens in there. And also the root has to be in the last row and last column of the E matrix. So from here, the root the root branches into two clones, two and three. The in E matrix, it is shown like that. Um, And similarly, we need another matrix Z, which will map the rows of the estimated copy number matrix to the observed copy number matrix because we put in individual constraints on the structure of the evolutionary tree and the estimated matrix. So finally, we try to minimize the evolutionary distance as well as the distance between the estimated and observed copy numbers. So this part has this thing has two parts. The first one here is for minimizing the difference between estimated and observed copy numbers, and the second one refers for phylogenetic cost, which is the cost of building the tree. And this is a regularization term. Lambda is a regularization parameter. I won't go into the details. You are welcome to look into our preprint. Uh, However, the goal is to minimize minimize the distance so that it follows the rule of minimum evolution. And finally, we imply dollar phylogeny constraints on to ensure a less restrictive model. Dollar phylogeny says that a mutation in a tree can be gained only once but lost multiple times. We ensure this by a new variable w, where wij is equal 1 if a mutation b occurs along the edge from I to j and 0 otherwise. And losses are allowed due to concurrent segmental copy number changes. So we applied the method to different simulation data sets. These are some of the results of our simulation data. In the x-axis, we have different number of cells from 20 to 100, and in the y-axis and left, we have root mean squared error of estimated versus true copy numbers. We compare our method with Scarlet, which is the existing method and very commonly used. Our method is shown in blue. So our results are better than Scarlet's. In the right side, we have the normalized RF distance in the y-axis, which is a common tree comparison metric for evolutionary trees. And we compared our distances with MEDIC2s and saw better results as well. However, one thing to note is that the better result is because we are using the different type of variants in one method. So we applied this method to uh, two high-grade serous ovarian cancer patients. The data set was collected from Funnel et al. And this tree had 133 single cells. We inferred a 5-clone tree. One thing to notice is that the clone branch from clone 3 to clone 1, it has an important SNV type mutation, which is TP53. However, the branch from clone 3 to clone 5, it has some important copy number type mutations, ERBB2, KRAS, NRAS, CCNE1, etc. So we conclude that the branch from clone 3 to clone 1 was mainly driven by SNVs, and the other branch was mainly driven by copy numbers. Imagine if we are only using one type of variant, we would have missed the second branch, clone 3 to clone 5 altogether. Now, we applied our method to a larger data set, with 1,283 single cells, and infer 7 clones in here. However, this tree shows some artifacts of our method, such as all the branches from the root has a distinct mutation SNV. It is the CDK17. We infer that this this is due to inferring insufficient number of clones in our method, and this is kind of an artifact of the method. So to summarize, we introduced the first phylogenetic tree reconstruction method that incorporates SNVs, CNAs, and SVs from single cell whole genome sequences. Our results show that different tumor clones are driven by different types of somatic variants, and to understand tumor progression in a larger scale, we need to incorporate all the variants. And we are currently extending our work to different type of single cell sequencing datasets, such as single cell RNA sequences. And another future direction can be to extend the method to complex structural variants, such as chromosynthesis events, which are chromosomal shattering. So with this, I'd like to thank my professor, Professor Russell Schwartz, and Shuetsong Fu, who for their great advice throughout this whole project, and the other members of my lab. The code is in Python and available in GitHub. The bioarchive version of the paper is also given there. Thank you.
We have time for a question or two. Please,
Yes.
Could you come up to the mic, please?
Great talk, thank you. I'm just wondering how likely those mutations happen independently in different clones. So if most of the mutations happen independently in different clones, then the type of analysis you are doing here probably is better called cluster analysis rather than phylogenetic analysis, because phylogenetic analysis would indicate that they follow the parent to descendant that the lineage is. Right, but if mutation happen independently in different clones, so this type of analysis probably again should be better called cluster analysis.
Great question. So we're thinking of extending the method for having like different getting different trees. So what we need is to set different constraints so that the roots are separated for this kind of event you just mentioned. But currently the method does not incorporate that. Thank you.